Greetings, listeners, and welcome to another episode of Genetics Concealed. Today I'll be talking about sickle cell disease, as well as blood genotypes relating to it. And I'll be talking about an article I recently read titled Researchers Use Supercomputer to discern whether molecules of life can be formed naturally in right conditions. At the heart of sickle cell disease lies a subtle yet impact mutation in the hemoglobin gene, known as HBB. This genetic twist orchestrates a cascade of effects, transforming the structure of hemoglobin and consequently the shape of red blood cells into the distinctive sickle form. But this genetic anomaly is not a random occurrence. It carries profound implications entwined with the threads of human evolution. The persistence of the sickle cell gene across multiple generations prompts a profound question. Why does this mutation endure within specific populations? To decipher this genetic enigma, we must embark on a temporal journey, peering into the annals of our evolutionary past and scrutinizing the intricate dance between genes and the environment. There are two places or ways in which it's thought that the gene for sickle cell came about. It's thought that it's either in an infant 25,000 years ago somewhere in Africa, or in three different African populations simultaneously at the same time. Moving on. In regions where malaria has been a relentless adversary, the sickle cell gene reveals a strategic advantage. Individuals carrying one copy of the mutated gene gain a unique resistance to severe malaria, a phenomenon known as heterozygote advantage, or the malaria hypothesis. This strategic survival advantage, offered by the presence of a single sickle cell gene, becomes a pivotal piece of the evolutionary puzzle. Zooming out from individual resilience, we explore the broader canvas of populations. Here, the selective pressures exerted by infectious diseases, like malaria, become entwined with the genetic adaptations that confer a survival advantage. Sickle cell disease emerges as a poignant example of evolution's nuanced hand, shaping our genetic landscape in response to the relentless forces of natural selection. But the tale doesn't end there. Sickle cell disease serves as a compelling window into the intricacies of human adaptation, shedding light on the delicate balance between genetic variations and environmental challenges. It unveils the complex negotiations that transpire between our genetic makeup and the selective pressures exerted by the diseases that have shaped our evolutionary journey. Now let's descend into the specifics of how one ends up with sickle cell disease and explore the blood genotypes that underpin its manifestation. Sickle cell disease is primarily a result of inheriting two copies of the mutated hemoglobin gene, HBS, one from each parent. The inheritance pattern becomes clearer when consider the various blood genotypes. Normal hemoglobin, which the affected state where both hemoglobin genes are normal. Sickle cell trait, individuals with one normal hemoglobin gene and one sickle cell gene generally do not experience severe symptoms. Sickle cell disease. The manifestation of sickle cell disease occurs when both parents pass on the sickle cell gene. So so normal hemoglobin AA, sickle cell AS, and sickle cell disease SS. Or you can add the prefix HB to both of those. Understanding these blood genotypes unveils the inheritance patterns of sickle cell disease, with a 25% chance of a child inheriting the condition when both parents carry the sickle cell trait. In an awe-inspiring experiment to determine whether the molecules of life could be formed naturally in the right conditions, the brilliant mind of UF PhD student Junzo Jue, I don't know the tones in that, so pardon me, harnessed the computational might of over 1,000 A100 GPUs on HiPerGator, one of the fastest supercomputers in the world. This technological symphony allowed him to conduct a monumental early Earth chemistry experiment, peering into the molecular dynamics of 22 million atoms. What emerged from this digital crucible were the fingerprints of creation: 12 amino acids, 3 nuclear bases, 1 fatty acid, and 2 dipeptides. A feat once deemed impossible due to computational constraints is now a tangible reality, all thanks to the convergence of HiPerGator's AI prowess and its vast GPU capacity. Professor Adrian Rotberg, um, the professor leading the Rotberg Computational Chemistry Group, underscores the transformative power of machine learning and AI. These advancements propel calculations at mind-bending speed, 1 million times faster than traditional methods. This quantum leap in scientific capability brings us to the brink of understanding the spontaneous emergence of complex molecules from the building blocks of life. Eric Dumans, the senior director for UFIT Research Computing, unveils the unique prowess of HiPerGator. Its capacity for large-scale calculations becomes the catalyst for breakthroughs in scientific frontiers. The collaboration between Dr. Reitberg's group and UFIT exemplifies the potential of AI and powerful GPUs in conducting data-intensive scientific simulations. The experiment not only witnessed the real-time formation of amino acids and intricate molecules, but also showcased the collaborative prowess of researchers and UFIT staff. Ying Zhang, UFIT's AI supporter, played a pivotal role in scaling compute runs, providing invaluable advice and accelerating data analysis. This scientific odyssey, from conception to results in a mere 7 hours, marks a significant step forward in deciphering the origins of complex molecules. The success of this computation not only highlights UF's capability to support hero runs or moonshot calculations, but also underscores the limited potential of science and technology in the pursuit of understanding our genetic world. Join me next week as we continue, as I continue to portray, to show the world the beauty of genetics. This is your host, Follow Deleke, signing off.